Genes are the blueprints of the human body. They instruct the body's cells how to make proteins critical to their function. Many diseases and disorders are caused by the faulty expression of proteins from defective or missing genes. Gene therapy is a technique that uses genes as a therapy. It aims to treat diseases by replacing, inactivating, or introducing new genes into cells to treat the underlying root cause of diseases. Bayer's gene therapy technology platform is built on the acquisition of AskBio, a pioneer in gene therapy. The basic idea is to add a functional copy of a gene which produces the desired protein in the patient's body. This protein can be, for example, an enzyme, a growth factor, or some other biologic. By inserting a functional copy of a missing or faulty gene into target cells, the functional protein can be restored, and the therapeutic gene then helps the cells to function correctly. Successful gene therapy requires three different components. In gene therapy, genetic material from outside the body needs to get inside the cells of the target organs. Currently, a validated approach to make this happen is through a delivery system called a vector. Think of a vector as a microscopic bus that transports cargo, the genetic material, to specific locations, the target cells. AskBio is using a modified virus as a carrier, the bus that I just described. You may have heard about AAV, an abbreviation for adeno-associated virus. The modified virus acts as a bus transporting the desired gene to a cell to restore the function of the defective gene. Viruses provide an ideal model for delivering gene therapy to the target location. Because of a virus's natural design, they are very effective at entering a cell. Delivery of the therapeutic gene is one of the key challenges in gene therapy. Precise tissue and organ targeting is essential to advance efficacy, mitigate likelihood of seeing immune responses, and maintaining overall safety of the treatment. Therefore, different AAV-based vectors are rationally designed to deliver specific characteristics. This enables efficient delivery with specific tissue targeting. It also supports the circumvention of neutralizing antibodies and increasing the applicability range of the therapies. While a number of viral blueprints exist, the choice of vector is based on characteristics such as duration of gene expression, the size of the genetic material it can deliver, target cells, and immunogenicity. Currently, viral vectors are the most common vehicle used in FDA-approved gene therapies. To further improve gene therapy, researchers at AskBio and Bayer are investigating different methods of delivering genetic material into cells. Beyond viral vectors, non-viral techniques are actively being studied for their safety and efficacy. The second component is a promoter. A promoter is a DNA sequence that sits beside each side of a gene and activates its transcription, the initial process whereby a protein is synthesized from the gene. Think of it as a switch that regulates gene activity. Promoters can be rationally designed to regulate the activity of a gene with a high level of selectivity. These are called synthetic promoters. Synthetic promoters can be included in the gene to be delivered with an AAV, allowing its activation. AskBio has previously acquired Synpromics, bringing deep expertise on this front. The innovative aspect of the technology we are working on with AskBio lies in an extended library of synthetic promoters and a proprietary data-driven design and bioinformatics approach. This enables the identification of more specific promoters, giving better control on the expression of AAV therapies, thereby improving safety and tolerability. As stated at the beginning, gene therapy inserts a functional gene into patient cells. This brings me to the third component of a gene therapeutic, a functional version of the defective gene called a transgene. The transgene is inserted into the viral carrier. This is the bus I described earlier, producing a functional copy of the gene of interest when it is at the target site. Here too, there's rapid scientific progress with innovations around transgene design to enable successful treatment. In summary, successful gene therapy requires an optimized vector, promoter, and a disease-specific transgene. Today, AAV technology has advanced to target a wide range of tissues and cells for the treatment of many genetic diseases. With a large number of ongoing clinical trials and multiple regulatory approvals in the last years, gene therapy is already a reality today for patients. The particularity of this approach is that it addresses disease at its source, the gene. This opens up the possibility for delivering disease-modifying therapies for diseases that have limited or no treatment options today.